Hi, welcome to AP Biology. I'm Gail Fizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. Lesson overview for today. We'll be reviewing topic 3.6, cellular respiration. This is in Unit 3. In fact, today finishes up Unit 3, and then the next lesson will be in Unit 4. We're halfway there. So cellular energetics is still in Unit 3. Anyways, today we're going to describe the processes that allow organisms to use energy stored in biological macromolecules. And we're going to explain how cells obtain energy from biological macromolecules in order to power cellular functions. Okay, so our warm-up today, which was our homework from our last lesson, which was photosynthesis with Ms. Kate. And if you are joining us for the first time, then I'm going to give a moment for you to jot down some thoughts in between after I read this question and before I reveal the scoring guideline. If you have your homework from last night, then go ahead and get that ready and we'll compare in just a minute. All right, in a certain prairie community, a dominant prairie grass species has recently been infected with a virus that disrupts one of the electron transport proteins in the chloroplast of the infected cells. Describe the most likely effects on cellular processes. Be specific as to which processes and molecules are most likely to be directly affected. So we have our first task verb of the day, and we're going to be describing. So I'll give you just a moment. If you're just starting with us, let me get my timer on. Okay. All right. So let's check your answers against the scoring guideline. So some effects that you may have included, you might have said that there was less ATP produced, less NADPH produced, inability to fix carbon via, via Calvin cycle without products of electron transport chain, and decrease the O2 production. All right, so let's get started with what we need to know about cellular respiration. I will go through this in detail, and I've kind of got to change up for the day of my usual routine, and I'll let you know when that change is about to happen. All right, so cells obtain energy from biological macromolecules in order to power cellular functions. So lipids, proteins, carbohydrates, all of those can be used in the metabolic pathways to make ATP. They just come in at different times perhaps, or in different ways. But carbohydrates is usually what our first thought about when we're thinking about making ATP. And that's the example we'll be using today. All right, so glycolysis is a biochemical pathway that releases energy in glucose to form ATP from ADP and inorganic phosphate. It also forms NADH from NAD plus and pyruvate. So glycolysis is happening in the cytoplasm of the cell. Okay, it's not in the mitochondria, it's in the cytoplasm. So here is our 6-carbon glucose molecule. And we're going to, our products will be 2 ATP molecules and 2 pyruvate molecules, and then 2 NADH. So how did we get that NADH? We had NAD plus come into the reaction, and then combined with a proton and 2 electrons, we're going to make NADH. And that NADH is going to carry the electrons to the electron transport chain further down the line in this process. Okay. All right, so pyruvate is transported from the cytosol to the mitochondrion where further oxidation occurs. So these are our 2 molecules of pyruvate that we just made in glycolysis. Now they can't get into the mitochondria without being actively transported in, but they are going to be, a reaction is going to occur, and the product's going to be 2 CO2s are going to be released. Again, we're making 2 NADH, and now we have acetyl-CoA that can go into the Krebs cycle. And here again, we have 2 NADHs. We had NAD plus the H, those 2 electrons, and NADH. And again, along with the NADH from glycolysis, the NADH from pyruvate oxidation they're going to go to the electron transport chain to drop off those electrons. All right, so now we're up to the Krebs cycle. Carbon dioxide is released from organic intermediates, and ATP is synthesized from ADP and inorganic phosphate, and electrons are transferred to the coenzymes NADH and FADH2. So if you'll take a look, you can see that here is our 2-carbon acetyl-CoA molecule, and they're going to combine with a 4-carbon oxaloacetate, and that's going to form this 6-carbon citrate. That's where the citric acid cycle name comes from. So in this cycle, we're going to produce 2 here and 2 here. So 4 molecules of carbon dioxide. And then we're also going to put electrons on NAD+. There's 2 NAD H's, 2 more, and 2 more. So there's our 6. So there's those electrons ready to go to electron transport chain. And then we're also going to use FADH2, okay, to carry electrons. It was built the same way. You had FAD, then 2 hydrogen, and then the 2 electrons are added. So now we have electrons on FADH2 and on NADH. Both of those have gained electrons, so both of those have been reduced. Okay, and then we can't forget our 2 molecules of ATP that are made also in that citric acid cycle. All right, electrons extracted in glycolysis and Krebs cycle reactions are transferred by NADH and FADH2 to the electron transport chain in the inner mitochondrial membrane. So here we're going, we have our glycolysis happened, okay, and we had these 2 NADH. We have our 2 NADH from our pyruvate oxidation, and then we have our NADH and our FADH2 from our Krebs cycle. And all of those are going to go to the electron transport chain. I know I have it very tiny right now. I'm about to make it larger so you can see what's going on. And so now that's on the inner membrane of the mitochondria. So that's where the reaction is going to occur. Excuse me. When electrons are transferred, here's a larger version so you don't have to try to figure out what was going on in that little one. All right, when electrons are transferred between molecules in a sequence of reactions as they pass through the electron transport chain, an electrochemical gradient of protons or hydrogen ions across the inner mitochondrial membrane is established. So here we have NADH. So where are we at right now? We're on the inner membrane of the mitochondria, okay? And this shaded area, this is why I've been obsessing over this diagram and the chloroplast one for the last 2 lessons, is because now we're going to use it. All right, so now we have NADH is going to, let me get a color I can see. All right, it's going to lose those 2 electrons, so it will be oxidized, okay? And then we have our NAD+ that can now go back to work in glycolysis or Krebs cycle. And then we also have all these protein complexes, and we don't need to know names or anything like that. This is more about what's happening, what the process is about, and what the process is for. All right, so we have these 2 electrons. They're going to be carried down the chain, and we'll talk about their future in just a little bit. But what's going on right now is this is going to go, there's hydrogen ions now, and they're going to go against the gradient to the other side, okay? So this hydrogen ion concentration gradient is being built in this intermembrane space, okay? So remember that was compartmentalized for an enzymatic reaction to occur. So that's how they're getting there. The NAD+, NADH is coming in and is leaving as NAD+. Hydrogen ions are getting pushed across the gradient, and the electrons are now on the membrane chain. Okay. All right, so when electrons are transferred between molecules in a sequence of reactions as they pass through the electron transport chain, an electrochemical gradient of protons, hydrogen ions, across the inner mitochondrial membrane is established. So let's take a look at this. There's more to this than just repeating what I just said. All right, so here's our inner membrane, and there is our reaction. Our hydrogen ions are building up within the intermembrane space, and they're doing this also over here. So why all of this? Why all of these loops and convolutions? Because we need what? We need increased surface area, because it's not just going on one place or two places here. This is all over this inner membrane. So this entire area that has been compartmentalized is now building up hydrogen ions. Okay. All right. Now, cellular respiration in eukaryotes involves a series of coordinated enzyme catalyzed reactions that capture energy from biological macromolecules. So now we can put it all together. We had glycolysis, and then we had pyruvate oxidation, and then we had the Krebs cycle, and then we have our electron transport chain. So now we can finally get to the point of why we're establishing that hydrogen ion concentration gradient. Okay, the electron transport chain transfers energy from electrons in a series of coupled reactions that establish an electrochemical gradient across the membranes. So let's talk about the word coupled. So we have this redox going on. So in case you need brushing up on your chemistry, so when you are reduced, okay, you're gaining electrons. When you're oxidized, you are losing electrons. So let's look at what's happening to NADH. Those electrons are getting off, right? So your NADH is being oxidized. And then look at this other end, and there's many other reactions that go along in the middle here, but look at our oxygen as our final electron. You can't see that. Our oxygen is our final electron acceptor in cellular respiration. So it'll be gaining electrons, so that would be reduction. So redox reactions are coupled. There's some gaining electrons, there's some losing electrons. All right, now electron transport chain reactions occur in chloroplasts, mitochondria, and prokaryotic plasma membranes. So if you'll take a look at this, here we are. This is, see this gray area? That's the gray area here. So this same electron transport chain is here on this inner membrane. And then if we look at the chloroplast, we have an electron, see this gray area? That's inside of the chloroplast in the side of the thylakoid, in the thylakoid lumen or the thylakoid space, whichever one you're calling it. So there's inside of that's where the hydrogen ion concentration gradient is forming. And then look in this bacteria. In bacteria, not a one size fits all of how things work. So let's just look at this model. And we have a cell membrane in orange. I just put it. And we have a cell wall in pink, and then this hydrogen ion concentration gradient will develop in between those two. And the same thing, that gray area would be with where the purple is. All right, so in cellular respiration, now we can get to why we're building up this hydrogen ion concentration. So in cellular respiration, electrons delivered by NADH and FADH2 are passed to a series of electron acceptors as they move toward the terminal electron acceptor, oxygen. So let's follow their path. So here was our NADH, and those two electrons were, NADH lost his two electrons. They're on the electron transport chain, and now NAD plus can go back and pick up more electrons and continue working. And the same thing will happen with FADH2, and then FAD can go do the same and pick up some electrons at the Krebs cycle. And in the process, on each one, we have hydrogen ions. They're going to be pushed against the gradient, and they're going to accumulate in this intermembrane space. Now, the electrons will eventually get off of this chain, and then they're going to bind with oxygen and then to hydrogen. And so their final electron acceptor is oxygen. Remember that later for an answer in an FRQ. Okay, so together, then that will form water. Now, why are we forming this hydrogen ion concentration gradient? What purpose is all of this that's happening in all of these organisms? That's because this amazing, let me get, what color do I want to write in? Okay, this amazing enzyme called ATP synthase is in all of these organisms that have these hydrogen ion concentration gradients. So the hydrogen ions, and this is, it's you need to find the video that you can see how this works, because they kind of go in and they go through this a little elaborate tunnel as they go from high to low. So they'll be moving this way, and the energy created from the concentration gradient is then the bottom part of the enzyme, it turns the bottom part of the enzyme. And as it turns it, it opens up the active sites for ADP and P, and then ATP is formed with that reaction that'll happen on those active sites. So that's pretty amazing. Now, we'll get to bigger diagrams, but in photosynthesis, the terminal electron acceptor is NADP+. It's right here, okay? And notice that NADP is in photosynthesis, it starts with a P, and then NAD, no P, is in the mitochondria. And then other molecules are used in bacteria, and there's a variety, but we still have basically the same. There's some protein complexes, and there's an establishment of hydrogen ions, and then there's the ATP synthase also. So I'll talk about photosynthesis. Let's do it on the next page because it's bigger. All right, the transfer of electrons is accompanied by the formation of a proton gradient across the inner mitochondrial membrane or the internal membrane of chloroplast, with the membrane separating a region of high proton concentration from a region of low proton concentration. So let's talk about what happens in photosynthesis. It'll be a review from yesterday, and then you can see the similarity of how this is, it's almost the same thing. All right, so we have sunlight, here's sunlight, there's sunlight. Let me give you your, let me orient you to everything. Here is photosystem 2, photosystem 1, ATP synthase, there's an NAD reductase, NADP reductase right here. You can barely see them. There are some mobile electron carriers that move in between these complexes. And so let's get started. So photon of light excites the chlorophyll pigment that's in this elaborate photosystem 2, and there's going to be some excitation, and those outer electrons are there'll be resonance, and that will make their neighbors respond, and they have electrons that get excited, so everyone gets excited, and then there will be 2 electrons that will leave the chlorophyll molecules and go to the electron transport chain. Well, in the meantime, there is a part of this elaborate complex is this enzymatic site where water will be split in the process when the sun hits this complex. So water is split, and we have our 2 hydrogen ions, and then we have 2 electrons, and then half of the oxygen gas. So, and it will bond with another oxygen gas and then leave. So, and diffuse out through the membrane. Anyway, so when this carrier, this purple that you can barely see right here, when it left this photosystem with these 2 electrons, it also picked up 2 hydrogen from the stroma and carried them to the next complex as well. And at the same time it moved those 2 hydrogen, there were 2 more hydrogen that were moved through the protein complex to end up with 4 entering the intermembrane space. And now I'm getting all mixed up. All right, so they entered the thylakoid space at that time. So now we have those 2 electrons still on the chain. Now the same thing will happen. Photosystem 1 will release, there will be 2 electrons that escape, and now there's no splitting of water at photosystem 1, and now the 2 electrons will replace those. 2 electrons end up to their electron acceptor, their final electron acceptor, NADP+, and then they become NADPH. And then that's going to head off to the Calvin cycle to run the dark reaction, or the light independent reaction. Okay, so hydrogen ion concentrations building up again, and then we have the same thing. It's going to go from high to low, open up that active site with that turn, and then form ATP, which will also go to the Calvin cycle. All right, so that's photosynthesis, and it looks just like almost just like what happened on the mitochondria. We have electrons moving, we have a hydrogen ion concentration gradient building, and then we had the synthesis of ATP from that hydrogen ion concentration gradient moving through that enzyme. And that's the pieces that you need to understand what's happening, not the name of photosystem 2 or photosystem 1, or you might need to know ATP synthase. I would know that one. But all these other members, you do not need to know their names. You just know how this all happens. Okay, in prokaryotes, the passage of electrons is accompanied by the movement of protons across the plasma membrane. So same concept. We're building a hydrogen ion concentration, and that hydrogen ion concentration gradient is going to drive the production of ATP. So all 3 examples that I just showed you do the same exact method. All right, so now there's some vocabulary that kind of goes with this process though. The flow of protons back through the membrane-bound ATP synthase by chemiosmosis, okay, drives the formation of ATP from ADP and inorganic phosphate. This is known as oxidative phosphorylation in cellular respiration and photophosphorylation in photosynthesis. So let's break apart what some of these words mean. Actually, the osmosis part of this is happening because this hydrogen ion concentration gradient is moving through. Okay, so that's the chemical part, and it's moving through from high to low, the osmosis, and that's forming the ATP, so that's called chemiosmosis, okay? Oxidative, what did we say oxidative meant? Oxidative meant you were losing electrons. And what does phosphorylation mean? That something is gaining a phosphate group, right? What gained a phosphate group? ADP plus P. So that was the phosphorylation part. And now we have a oxidative, we lost our electrons, and there's many other reactions that happen along this line that's called oxidative phosphorylation. The difference in photophosphorylation has to do with the photons of light, with the energy coming from that first. All right, so now we have been through cellular respiration that has to do with having oxygen. Now we're going to talk about fermentation, and fermentation allows glycolysis to proceed in the absence of oxygen, okay, and produces organic molecules, including alcohol and lactic acid as waste products. So if you look at everything in this area, okay, well, I kind of should have got that. That's glycolysis. In glycolysis, we made 2 ATPs and 2 pyruvates and 2 NADHs. So same thing, glycolysis is part of fermentation of each part. But in alcohol fermentation, now we can't go into the mitochondria, can we? No, because we don't have oxygen. So what's going to happen is that this pyruvate is going to end up taking back those electrons, so it will be reduced. First, it'll lose 2 carbon dioxide molecules and become acetaldehyde, and it gains back those 2 electrons, so it's reduced. NAD can go back to work to make 2 more ATP, because that's where we're getting them from in fermentation. That's the only place we have to get ATP from. And then our end product here is a molecule of alcohol, ethanol. So what are some examples of this? Bread and we're making bread, and you can make wine and beer. And we actually made bread in my classroom this year, and the smell goes down goes down the hall, and it smells so good. All right, and now the other type is lactic acid fermentation. And look at this. See, we're still the same. Glycolysis is still the same. We have 2 ATPs, we have our pyruvate, we have no O2. So that's, so we're not going into mitochondria. Now, this would be in, well, we can do this in eukaryotic cells. So anyway, no oxygen to go into the mitochondria. Now I'll give you an example in a second. First, is there CO2 with this one? No CO2 is a waste product of this. So what happens is this pyruvate gets back those 2 electrons and then is going to form lactate. So our NAD can go back to work and make more ATP. So this can run for a little while. And speaking of running, if you remember any time that you were running and you had pain in you got out of breath and you had pain in your liver area, for example, and you had to stop and breathe for a while, that was the lactic acid. It's a toxin and it's building up. And that means that you're not running on ATP that was from your mitochondria. Now you are into getting the ATP from your lactic acid fermentation from glycolysis, and then you're going to only be producing this other toxin, and that's what's causing the pain. So you have to stop, you have to breathe some more, you have to breathe enough to remove the toxin from your cells, and then you have to breathe back enough to get a storage again of the oxygen before you can get going again. Now, respiration and fermentation are characteristic of all forms of life. All right, so the conversion of ATP to ADP releases energy, which is used to power many metabolic processes. So this is very simplified, but here's our ATP molecule, and there's our phosphate groups. I lost my bond somewhere along the way. All right, so there's some high energy bonds between these. Now, when this is ADP, and this is P. So when this bond is broken, the energy is released, and that energy will fuel a cellular process, okay? It's a very small molecule with a small amount of energy, nothing like the big glucose molecule, and we'll talk about that. All right, so now let's check for understanding. In fact, we're going to talk about that right now. All right, so we're not really going to need these diagrams, but you see we have, here is, just for review, there is our glycolysis. Then we have pyruvate oxidation, Krebs cycle, and our electron transport chain. So the question, a researcher estimates that in a certain organism, the complete metabolism of glucose produces 30 molecules of ATP for each molecule of glucose. The energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. Yes. All right, so the energy released from the hydrolysis of ATP to ADP and inorganic phosphate under standard conditions is 7.3 kilocalories. Now calculate. So now this is our first calculate for the day. The amount of energy available from the hydrolysis of 30 moles of ATP. So what is this? 30 times 7.3. And that's 219. And so we did that. Now calculate the efficiency. This is where the tricky part comes in, because sometimes kids don't know what that means for them to do. The efficiency, what it means is if we had this much available, if we had 686 available and we only used 219, that part is the efficient part. So what you do is you take the 219 that we were able to use and divide it by 686. And so my calculator says 0.319. So we will say it has a 32% efficiency. Now, the next part of this where there's describe. So we've had calculate, calculate, and describe. What happens to the excess energy that is released from the metabolism of glucose? So we started off with this much energy and we only have this much we can use. Where'd the rest go? I'll let you take a moment to think about that. Jot down an answer, and then in about 4 seconds, we will, I'll reveal the scoring guideline for this. Okay, so did we say 219? Yes, we did. 32%? Yes. And did you jot down released as heat? You could have said increases entropy, but this would probably have been my go-to if I was answering that question. Okay. Now, instead of more multiple choice practice and things like that, I had a request from, and here's a shout out to Patrick from Massachusetts. And Patrick wanted to start practicing in a large FRQ, and he asked if we could make that happen this week. And the timing was perfect, because if you have seen this practice icon, these are practice questions that are modeling what's going to happen on the AP exam. So in your AP classroom, your teacher does not have to assign this to you. They're there for you to, she can, but they are there for you to practice on your own if you wanted to. So that's where I got this question. And so let's start. And I want to do something. I want to write down that this is what my timer says right now. Okay, so I'm looking at my diagram. I see I have glycolysis, and we're producing NADH, and I use it in the electron transport chain when I have oxygen. I use it in fermentation when I don't have oxygen, and the end result of using the electrons from the NADH that carried was that I get NAD plus to go back to glycolysis. So electrons from the oxidation of glucose and other nutrients by glycolysis and the Krebs cycle are added to the electron carrying compound NAD plus to form NADH. To meet the continuous requirement for NAD plus by cells, NADH is reduced back to NAD plus through the electron transport chain in the presence of oxygen, or through fermentation in the absence of oxygen. All right, so this is the question. All right, so I'm going to write after I read. All right, mitochondrial ND5 is a mitochondrial gene that encodes a subunit of NADH dehydrogenase, the enzyme that catalyzes the initial oxidation of its substrate NADH to NAD plus and H in the electron transport chain of mitochondria. Let's stop right there so I can write myself a note if I was answering this question. Okay, there's this, and that's a gene, and it makes what? It's an enzyme that's going to take apart NADH to NAD plus and H. Okay, because I got to remember that, and I don't want to go back and read it. Okay, so a mutation in mitochondrial ND5 is associated with a rare genetic disorder that results in a buildup of lactic acid in the body. So we're going to have, because we have this mutation, we're going to have an increase in lactic acid. A researcher hypothesizes that the mutated NADH dehydrogenase has decreased activity, but is not completely non-functioning, and that by increasing the pool of NADH in cells, the activity of NADH dehydrogenase will increase. To test this idea, the researcher tested a group of individuals with this disorder with a vitamin that is similar to NADH and measured the concentration of NAD plus and lactic acid in the blood over the course of 20 weeks. The results from a representative individual are shown in figure 2. So let's go to figure 2. All right, so I have a graph, and I have concentration of NAD, and then I have a graph with concentration of lactic acid. Both are over 20 weeks. They're over weeks. Okay, and I see a peak at week 5. And what about week 5 here? It's a low point, but overall the trend, I see a decrease going down of the trend. And this trend went up and then down. Okay, so describe, we're going to do A, describe the pattern of inheritance that is most likely associated with a mutation in the MTND5 gene. Okay, so that's one thing we need to do. Explain why individuals are not typically heterozygous with respect to mitochondrial genes. So all of a sudden, there's this information that we need to have in this question that had nothing to do with what we thought we were going to be talking about. So a long FRQ is not going to just talk about one unit. So like we're in cellular energetics and that's 3, but this just brought in unit 6 material. And that's the way a large FRQ is going to work. It's going to be a combination of unit concepts plus also science practices. So let's answer, if you jotted that down, let's answer that in just a moment. I'm going to tell you what I think. I'm going to think that the mitochondrial gene has something to do with maternal. Okay, that's where we got it. If we get any of the mitochondrial, maternal inheritance, and why are individuals not heterozygous? That's because there's only, that's a mitochondria, there's only one copy of the DNA. So hold that thought, and I hope you and I score at the end of this. All right, so B, identify a dependent variable. So we have to identify, so we're just going to write down the words measured in the researcher's experiment. So let's go ahead and do that. What can we say? The dependent variable, we could have concentration of NAD. We can also have concentration of what? Lactic acid. So let's write that down. NAD and lactic. Okay. Identify, here we have another identify, one control that the researcher could use to improve the validity of the experiment. Justify the researcher analyzing blood samples at many intermediate time points instead of only at the beginning and the end of the 20 weeks. So we have 2 things to do. We've got to identify a control, and then we've got to justify. So controls, I always write 3 things about a control. I say the control has no treatment. I say that the control shows what's normally happening without the treatment. And then the control also shows that the treatment is responsible. Okay, those are 3 things that if I'm writing about a control, I'm always saying those things. Whether it's needed in the rubric or not, I'm always saying those things just in case. Now, justify, remember we have a claim and evidence and a reasoning when we're justifying. So let's take a look, and why would we need to take this? What are we getting from taking these readings instead of just here and just here and just here and here? We're getting what? We're getting a trend, right? We're getting a trend. All right, so we can claim that we're getting a trend, and that would be, we could use our numbers if we wanted to, and then we could reason it out with, this would give us more data to examine or something like that. All right, I mentioned the planning stages. All of this is something I do to get started with writing, because the writing's the easiest part. The planning's the hard part. Okay, now C, describe the relationship between the concentration of NAD in the blood and the concentration of lactic acid in the blood during the first 5 weeks of treatment with the vitamin. Okay, so what's happening with these 2? I need to get rid of some of these lines. So we could say that did it say at 5 weeks to look at it? Yes, okay. So we could say at 5 weeks that as NAD is increasing, what's happening at the lactic acid? We have decreasing, right? Okay, we can say that. And then we also need to calculate, here we are calculating again, the average rate of change in blood NAD plus concentrations from week 5 to 17. Where'd my graph go? All right, so let's look at week 5. Here's week 5. Here's week 5. And we have 300. Oh, we don't need that one. Never mind. we have 300, and then it said week 17, so that's right here. I have the ability to have a little cheating device right here to help me, so I can see where this is going to be level. Okay, so it's not at 50, it's above 50, it's below 75. So let's say it is at 60, okay? If this is a 300 and this is at 60, then how much did it change? It decreased by 240. Now, over what period of time? 5 weeks to 17 weeks, so that's 12 weeks. So what's 240 divided by 12? It's 20, and then we go micromoles per liter, and then what are these? These are weeks. Okay, so that's our answer. Now, what did we just do here? We did simple calculations that did not require a calculator. So I would get, this could be something that would be easily testable, is finding this kind of rates are really easy to do without a calculator. Let's just keep that in mind and get really good at it. All right, so now let's go to D. The researcher performed a follow-up experiment to measure the rate of oxygen consumption by muscle and brain cells. Predict the effect of the mitochondrial ND5 mutation on the rate of oxygen consumption in muscle and brain cells. So let's think about what happened. So we have this gene and there's a mutation in it. And so what were we not doing? We're not making that enzyme that took NADH to NAD. So what didn't happen? So in our electron transport chain, electrons did not happen. So they didn't travel down and they didn't get off on oxygen as the final electron acceptor. And so then oxygen wasn't needed. So what do we think is going to happen with the oxygen consumption? It's going to decrease. And then all those things I just said by drawing it out and thinking it out would be my justification. The researcher had hypothesized that the addition of the vitamin that is similar in structure to the NADH, I'm gonna give you a hint, okay, would increase the activity of the mutated NADH dehydrogenase enzyme in individuals with the disorder. Explain how the vitamin most likely increased the activity of the enzyme. I'll give you just a moment to think about that, and because I'll let you do at least one part all by yourself. I'm actually just modeling this so you can see how to plan, because that's going to be critical. Okay, so now let's look at the scoring guidelines. So this is for A description that the mutant allele is only inherited from maternal. Did I get that point? Yes. And then is a mitochondrial gene, and mitochondria only carry a single unpaired chromosome. Okay. And then on part B, an acceptable dependent variable. Acceptable dependent variables include the concentration of lactic acid in the blood and the concentration of NAD. We got those straight off of the dependent variable on the graph on the y-axis. And then identification of the control as measuring NAD plus and lactic acid, all right, in a group of other affected individuals treated with a placebo. What's a placebo? A placebo means they're not getting the treatment. I wouldn't have thought of saying that. I didn't read enough into that question, so I'm not sure if I would have gained that point with all my control talk, but maybe so. All right, hold on just a minute. Let me get rid of something. The justification that by collecting more data, the researchers will see a more accurate trend. All right. Now C, a description that the concentration of NAD increased and the concentration of lactic acid decreased. Yes. A calculation of 20. We got that. Now, the prediction that mutation will decrease the rate of oxygen consumption by the electron transport chain. The justification that the mutation likely inhibits the rate of oxygen consumption because the electron transport chain will be less efficient, so less oxygen will be required as a terminal electron acceptor. And then this part, and I hope you said this, the explanation that the enzyme had decreased activity but still functioned Increasing the amount of substrate should increase the amount of product produced. Because the vitamin was similar to NADH, it could bind to the active site and increase the substrate concentration. How about that? All right, so let's look at this time, 42:53. All right, 29:31. All right, so 29:31 is when I started. And I started reading and I started planning. And that was 13 minutes. So now I have 12 minutes. It's all figured out to put this into some decent sentences that make sense and then upload. So this is so doable. If you're prepared and you have a plan and you do your planning, this moment of being 25 minutes, you should know you're going to have plenty of time to get that job done. All right, so debrief and summary. Students, you have access to optional free response questions to practice demonstrating your knowledge and skills for the exam. Look for the practice icon. This is my new best friend. These do not have to be assigned to you by your teacher for you to be able to practice, and they are similar in design to the questions on the exam. They won't be the same content or any same topic, but they will be they're modeling what it's going to look like, and one important thing for you to do is model what your time frame is going to take, okay? Start practicing with that timer. Get one running on the computer in front of you to go ahead and make you nervous that the clock is ticking, because I just did this planning with you in very few minutes and live on YouTube's in a very stressful condition. So you can do it too. This bottom blurb is for your teachers. Be sure to use AP Classroom to help you. I use AP Classroom topic questions to warm up. I see my kids online every day. We start with AP Classroom topic questions to warm up. Then I talk through a topic with them, and then I have them watch parts of the videos. They have to have me making them watch me. And then we pull it all together, and then they answer FRQs that are within the video that we watch. So the people at AP Central at College Board are working so hard to give all these tools to these kids to help them. I've never seen a group of people so determined to make that happen. All right, so your homework tonight is a question. This is kind of like a lab that you might have had in cellular respiration unit. I had peas and corn and beans and all of those things in my room, and snails and roaches and worms. Anyway, let me read this to you so I can get you out of here. An agricultural biologist was evaluating 2 newly developed varieties of wheat as potential crops. In an experiment, seedlings were germinating on moist paper towels at 20 degrees for 48 hours. Oxygen consumption of the 2-day-old seedlings was measured at different temperatures. The data are shown. Okay, so here's your graph. You have 2 varieties, 2 temperatures, and your job is, your homework, explain the relationship between metabolism and oxygen consumption. Discuss the effect of temperature on metabolism for each variety. So 2 task verbs are included in there. All right, so that's your homework. Have that ready for Monday or the next lesson. All right, so if you don't have internet or a device, please, please, please contact cb.org/tech to let them help you. And you need to do this by April 24th. Also, big on your list right now, check your AP account email, make sure it's right, because they're going to send you a test ticket. All that information is going to roll out to you the week before the test, and we'll be here to help you with that as well. And thank you for watching. Next session will be with me also. It's going to be self-communication. Yay, we're on to Unit 4. And so that means we're halfway done. All right, thank you. Bye.